Hello everybody, and welcome to the third video in the meiosis unit. And in this video we're going to be discussing genetic variation. So in sexual reproduction, we already know that the offspring that are produced are genetically unique from each other, and they are also genetically unique from the parent, meaning that they have different traits than each other and different traits than the parent. So this provides an advantage to a species evolutionarily, because I want you to imagine if everyone in a species were exactly the same, and they had the exact same genes. That means they would all be equally as likely to be caught by a predator. Or if there were a disease that you might catch based on what genes you have, that means everyone in that species could catch the disease because they all have the same genes. So it's important to have variation because that allows some individuals to survive even when others do not. So genetic variation comes mostly from meiosis. So we're going to be looking at three different sources of genetic variation in this video. So the first one is something called crossing over, which happens during meiosis. The second is called independent assortment, which also happens during meiosis. And then the third is random fertilization, which happens during the fertilization step of sexual reproduction. So first, a little bit of a reminder of some vocab. We're going to be using the phrase homologous chromosomes in this video quite a bit. So remember, in a diploid organism, chromosomes come from pairs. So humans, we have 23 pairs of chromosomes. One set of 23 came from our mother and one came from our father. Now the two chromosomes that carry the same genes, that carry the same traits, those are called homologous chromosomes. So, this might be the chromosome from your father that carries the gene for hair color, and this might be the chromosome from your mother that carries the gene for hair color. And whether they're in their unreplicated form or their replicated form, these two chromosomes, 1, 2, are a homologous pair. Also, these are also a homologous pair because they had the same traits. So first, if we look at a regular old picture of meiosis, this is nothing fancy going on here. Remember, during metaphase 1, the homologous pairs pair up at the metaphase plate and then separate. So then at the end of meiosis 1, we have haploid cells that have one of each homolog in each cell. So we've separated the homologous pairs, and then these cells divide again in order to separate the sister chromatids in order to get non-replicated chromosomes, giving us 4 daughter cells at the end. I want you to remember this picture because we're going to be referring back to it quite a bit during this presentation. So the first thing that happens during meiosis that can generate genetic variation is called crossing over. Now this happens during prophase 1. We actually mentioned it in the steps of meiosis video, but I told you, hey, don't worry about this now, we'll come back to it later. And remember, one of the things that happens during prophase 1 is the homologous chromosomes find each other and pair up. So this is actually where they first pair, is during prophase 1. Remember, we have replicated chromosomes at this part because S phase has already happened. And during crossing over, during prophase 1, while chromosomes are paired, something kind of funny happens. And that thing is called crossing over. So if you look at this picture here, what happens is two sister chromatids, one from one chromosome and one from the other chromosome, will break off and swap portions. So now I literally have a piece from what was the blue homolog is now on the white homolog, and vice versa. Okay, so sections of homologous chromosomes that code for the same trait, they're swapped. So maybe the C gene was the hair color, where the white C is blonde hair and the blue C is brown hair. You can see that now they got swapped in those two chromosomes. And what this does is now it makes 4 different versions of that chromatid. We have the all white version, the all blue version, and then the one that's like half white and a little bit blue, and the one that's half blue and a little bit white. So what this does is it mixes up genetic material from the individual's mom and dad to create basically new combinations. Whereas before little a, little b, and little c were all together, and big A, big B, and big C were all together, and now we have different combinations. We have little a, little b, and big C together right here, and vice versa right here. So this is creating more variation. Okay. So therefore, if I have normal meiosis over here with no crossing over. And by the way, meiosis always has crossing over, so we're calling it normal, but it's not really normal. So we have meiosis with no crossing over on this side. You can see there are 2 cells with a big blue chromosome and a little red, and 2 cells with a big red and a little blue. Whereas where we have crossing over, every single daughter cell is unique. So crossing over has given us more variation than we would have gotten without it. So the second way we can generate genetic variation during meiosis is via this idea called independent assortment. And this takes place during metaphase 1. And let's look at these pictures first. So I want you to imagine if we only had 2 pairs of chromosomes, so 4 chromosomes total, there are 2 ways these chromosomes could line up during meiosis. By the way, in these pictures, let's pretend the blue chromosomes represent the ones from your father and the red represent the ones from your mother. They could line up with the paternal chromosomes on the same side and the maternal on the same side, giving you all blue or all red offspring. Or you could end up with 1 blue and 1 chromosome on one side and 1 red and 1 blue on the other, which would give you a big blue and little red and a big red and little blue. Because each of these possibilities is equally likely, that means sometimes when our bodies undergo meiosis, we'll get this version, and sometimes we'll get this version. So this idea that homologous chromosomes line up on the metaphase plate 2 by 2, and it doesn't matter which chromosome in the pair is on the left and which is on the right, that means you get different combinations of chromosomes each time you do meiosis. Now this is further complicated by the fact that we don't just have 2 sets of chromosomes like this picture shows here. In reality, in fact, we don't even have 3 sets like this picture is showing here. So you can see if you have 3 sets of chromosomes, there is even more ways they can line up, right? You could have the blue all on the same side. You could have the big red and the medium red on the same side. You could have the big red and the small red on the same side, right? So really, we actually have 23 pairs of chromosomes, which means there are a ton of different ways that our chromosomes could line up differently, giving us different egg and sperm cells via independent assortment. And there are so many different ways that egg and sperm cells, it's very, very unlikely that 2 sperm or 2 egg cells that you make are identical. So independent assortment makes each individual's gametes genetically different because of the order in which the homologous chromosomes ally. So this basically means one sperm that you make versus another sperm cell that you make are not going to be the same, and this is going to make it very unlikely that your offspring will be identical. So I told you there are a ton of different ways that our chromosomes can line up to give us unique gametes. In fact, there's a formula to calculate this. So you can calculate this with the formula 2 to the number of homologous pairs. And because there are 23 homologous pairs in humans, that means using independent assortment alone, we can make 8,388,608 different gametes. And this is only using independent assortment. So that means there is a 1 in 8,388,608 chance that 2 of the sperm cells or 2 of the egg cells that your body produces will be identical. Now, if you add in crossing over to that, which happens all the time in every meiosis, you can kind of see why no 2 sperm or no 2 eggs from the same person are going to look the same, which is why siblings never really look the same. Kind of cool. So those 2 ways actually aren't the only ways you can make genetic variation. Also, what we call random fertilization also adds genetic variation to the population. So we already know that meiosis makes the gametes, makes the cells that are needed for fertilization to happen during sexual reproduction. And we're going to define fertilization here as when the nucleus of a sperm cell fuses, aka combines with the nucleus of an egg cell to make a zygote. We already know that word, okay? And we already know that human cells have 46 chromosomes because they're diploid. 23 came from each gamete. So there's a cool picture over here. So this is actually a picture from a microscope that's been colorized. So the photo was taken in black and white and color was added after the fact. But you can see here's an egg cell. And here's the little sperm cell, and the sperm is penetrating the egg cells so that their nuclei can combine to do fertilization. So now, I want you to imagine if you had a man and a woman who were going to mate here, the man can make 8 million plus different sperm cells, and the woman could make 8 million plus different egg cells. So therefore, the fact that the sperm that fertilizes an egg are genetically unique due to crossing over independent assortment, and it's pretty random which sperm will randomly fertilize which egg, that means there are tons of different combinations of which sperm and which egg could combine from these 8 million sperm and these 8 million egg. And this makes the number of different zygotes two people can produce absolutely skyrocket. So again, if you think of the fact that your mother could make 8 million different eggs and your father could make 8 million different sperm, and that one of each we're going to combine, you can kind of see why you don't look like your siblings, because the likelihood of the same two happening to combining is pretty low. Okay, so now once the genetically unique zygote is formed, then it will undergo mitosis over and over again to create a baby, to create an offspring. So finally, to summarize, sexual reproduction produces genetic variation among offspring, making them genetically unique. That's pretty important because you do not want everyone in your species to look identical, because otherwise, one disease could wipe everyone out at the same time. You want variation, which allows some people to live and some to not, to guarantee that your species lives on. And this genetic variation comes from 3 sources: crossing over during prophase 1, independent assortment during metaphase 1, and then random fertilization during fertilization. So that's it, and I'll see you in the next video.